I forgot. So I think we have to talk a little bit again diabetes at the end. So people who are living in the town areas, they are more affected because diabetes is mostly lifestyle related disease. It's lifestyle related disease, uh, type 1 diabetes, right? And type 2 diabetes, there are two diabetes, type 2 diabetes. So we are talking about type 2 diabetes. This is common. These people, insulin level low, or the cells are not responding to insulin. Now, what is the role of glucagon? Let's assume we are not going to eat a meal. We're going to skip the lunch. If we skip the lunch, our glucose level decreasing because body need energy, the blood glucose level decrease. When the blood glucose level decrease, glucagon hormone release from the pancreas islet of Langerhans. Glucagon. So if there is a blood glucose level decrease, then the glucagon is released from the pancreas. Then glucagon breaks down glycogen into glucose. Now you see, glucagon is the hormone which initiate event to break down glycogen into glucose. Then the blood glucose level again increase. So this is the role of glucagon and insulin in pancreas. All right, so summarizing, last thing, pancreas secrete 5 hormones: lipase, trypsin, chymotrypsin, amylase, and nucleases. One chemical bicarbonate ions under the influence of secretine, and two hormone, glucagon and insulin. Insulin decrease blood glucose, glucagon increase blood glucose level. People sometimes used to call this fed fed state. Fed mean feeding. After we eat, fed state, and this one is a fasting. Fasting means we skip the meal, it is fasting. Fasting stage, glucagon is active, fed state, insulin is active. And we also learned the main structure, basic structure of the pancreas, made out of lobules. Lobules made out of acinus, collection of acinus, and especially the islet of Langerhans is the endocrine part of the pancreas. Exocrine and endocrine part of the pancreas. Okay, little bit we learn regulation of glucose by insulin and uh, glucagon. So, do you have questions? Anything to ask? Okay, then we can move to the next lesson in the tute. Next lesson is I can't remember. It should be the liver. Yes. Next topic is liver. Okay, so we start liver. Anyway, you see, my computer is getting very slow because battery life is now 16%. But in few minutes, I should get the power. That's why it is slow. All right, now liver is a very important organ in digestion, right? So if we look the gross arrangement like in pancreas, how it looks like, the liver has a connection directly with the intestine, like a small intestine. So this is the small intestine, and the small intestine connect with the liver, the called portal vein. Portal vein connect to the liver, and liver also get a direct blood supply from the heart. This is the hepatic artery, right? So it's very special, right? So it has two ways. Blood is coming from two ways. One is coming from intestine, one is coming from the heart. But it has only one connection to the heart, then it's going back through this hepatic vein. So hepatic artery bring the blood, portal vein also bring the blood. And there's only one connection out of the liver, hepatic vein. And there's another important connection. The bile duct is again going to the intestine. Bile duct, portal vein, hepatic artery, and the hepatic vein. So now interestingly, this is going out, this is also going out of the liver, and this is coming to the liver. Right. Now, if we look the gross appearance is what we can see by eye, not the microscope. The liver is again divided into four lobes. It's not lobule, lobes, right? Lobule are the small structures, lobes are bigger structure, liver lobes. So we can find four big areas of the liver. It is separated. It is not a very single organ like things. We can find four separate units, make this one structure, the liver. And we always use this term hepatic, hepato. This is related to the liver. If anything coming like hepatic, hepato, it's a liver related. Understood? Hepatic artery, portal vein, they bring blood to the liver and take blood out by the hepatic vein. And this blood is oxygen rich, oxygenated blood, and this is also again oxygenated blood plus high in nutrition, because all the digested product from intestine moves to the liver first through the portal vein. When the hepatic artery and portal vein both have the same type of blood, oxygenated blood, they're going to mix up in the liver. So they can mix up in the liver, and then oxygen delivered, nutrition delivered, and then the deoxygenated blood move to the vein side to the heart. So this is the again gross appearance of the liver. And I miss again a little bit of things. The bile duct is also connected with the gallbladder, because bile is temporarily stored in the gallbladder, and then only it's moving when there's a requirement. The bile is moving to the intestine. That's also regulated by cholecystokinin. You can check previous note. I mentioned what is cholecystokinin, and it regulates bile secretion. Now, like in pancreas, we're going to look what is the microscopic appearance of the liver. Microscopic appearance of the liver. Your battery is running low. I don't know if I don't get the power now. Okay. Should come now. It's 7. Okay, I got the power. So these lobes, 4 lobes here. These lobes made out of collection of lobules. Collection of lobules. Now in pancreas, we only see lobules, not the lobe. Now liver, we can see 4 lobes. Lobes are made out of lobules, right? So if this is the liver with 4 lobes, 1, 2, 3, 4, and we can find tiny structures like this to make the lobules. I think hepatophyta. So hepatophyta has a liver-like appearance on the surface of the leaves because of these tiny lobules connected to make the lobe. And we take a look into these tiny lobules. These small shapes, the lobules, made out of very special unit called the hexagonal shape. 2, 3, 4, okay, like this. 1, 2, 3, 4, 5, 6. So these are the typical shape, and they are made out of this kind of a hexagonal shape functional unit. So this is the lobule, hexagonal shape. Okay. So hexa is a six, one side, two side, three side, four side, five side, six side. So liver is made out of lobes. Lobes are made out of lobules. Lobules are made out of hepatocyte. So remember the order: lobes. Lobes are made out of lobules. Lobules are made out of cells called hepatocytes. Lobes, lobule, and lobule are made out of hepatocyte. So this is the arrangement of the liver. Lobe, lobules, and lobules are made out of hepatocytes. Hepatocytes are tiny cells like this. Okay, so again summarizing, the liver get hepatic artery, portal vein, both bring oxygenated blood, and the hepatic vein take out the deoxygenated blood. And bile duct is also leaving out of the liver. Okay, now if we take this hexagonal shaped lobules, they should also receive these blood vessels, right? So for example, the hepatic artery should come to this hexagonal shaped lobule. But when it comes to hexagonal shaped lobule, the blood vessel size should be reduced, because you can understand, when it comes from the heart, the blood vessel has a bigger diameter. So we call that artery. We call it artery because it has a bigger diameter. But the artery is now branching, because artery is a big one. It cannot directly supply to these lobules. So this artery is branching, and this branch now moving to this hexagonal shape area, and they are called, when it branch, we call it arteriole, right? So we call it arteriole. Hepatic artery branch into arteriole, and then arteriole, when it come to the cell, it become further reduced to become capillary. So this should further become to make the capillary. Since we haven't learned the circulation, we can understand this division: artery, arteriole, capillary. Based on the diameter of the blood vessel, we can name artery, arteriole, capillary. Then what we can find inside is the capillary, because they are very close to the cells. And similarly, we can see the portal vein. Portal vein is also branching like this, and then it comes to the capillary. In that case, it is portal vein, and the small branch is venules. Small branches are capillary then. So portal vein, venule, and same capillary. Okay, now we should expect these two blood vessels in this hexagonal shaped area. Now after that, what is going out, that we can find in this, in the middle, because the blood flow is this direction. Blood is flowing from this direction to the center of the hexagonal shaped lobule, and this is the, it is called central canal, and central canal make the hepatic vein. So it make the hepatic vein. Right, still one more thing to come, bile duct. So bile duct should come out of the hexagonal lobule, because cells in this area produce bile, and bile is also moving this direction. So we should also find a bile duct, tiny bile duct here. This is coming towards, this is also coming towards, this is going out, and this is also going out. So it is very special organ, liver. To understand it, we need a little bit of patience. We have to learn a little bit of patience to understand the structure of the liver. Okay, now hope you understand this one well, and then you understand this one well. Now we are coming the detailed structure of the lobules. Okay, when you come into the detailed structure of the lobules, these hepatocytes, right? I take just to make it easy, one area like this. This is the portal vein. This is the hepatic vein, branch of the hepatic vein, branch of the portal vein, and at the same time, we can also find the bile duct. So at one corner, let's say this is one corner by drawing this. This is one. Like this. The hexagon shape, the lobules. At one corner, we have these three structures. The hepatic vein, branch of the hepatic vein, branch of the hepatic artery, no, hepatic portal vein, right? Portal vein, hepatic artery, branch of the hepatic artery, and the bile duct. So it's a collection of these three ducts we can find each corner, right? So here it's the same. If you look, again we have the same thing in this corner. Okay, now the hepatocyte has very special arrangement. The hepatocyte make columns like this, and there is another column like this, and there's another column like this. Okay, now if you look the space here and also this space here, is the branching of both the portal vein and the hepatic artery. Portal vein and the hepatic artery both are branching into capillaries in this area. So I try to draw the cells of the capillary like this. And there's a very special thing we can notice. They are not really making a continuous tube. When it portal vein and hepatic artery branching between these hepatocytes, they are broken up, like the cells are broken up, and they mix both portal vein and the hepatic artery. The blood is mixed. Blood is mixed, and there is a area which is open up, like they are not the really closed. The blood vessel is not really closed structure. It has many gaps. So this kind of a structure is called sinusoid. Between the broken structure is called hepatic sinusoid, which is the collection of portal vein and hepatic artery blood, venous blood. Right? Now there is a problem. The problem is if intestine, the portal vein bring the blood. If the blood contaminated with some microorganism or whatever, they can escape from this place. So they can enter into the system because there is a chance the gaps. Otherwise, you will learn in the future. Uh, it is extremely dangerous if some disease causing agent enter into the blood. Our body is protected many ways that they can't enter into the blood. Why is that? Imagine through the blood it can travel to the brain. Through the blood it can travel to the heart. If they go to the heart and the brains, these are the vital organ, the person can die. It is just like the terrorist attack to the Pentagon of USA. The Pentagon is the operating center for all these things, but if they can move into that place, it's very dangerous. Because human body is made like that way that they can't enter into the body. It could happen in many ways, like even they can enter into blood from this place. Like if there is a infectious agent, they can now move into the blood because the space is big. Because of this, both breaking into a broken blood vessel called sinusoid. That's a mixture of these two blood, and it is dangerous. That's an entry point for pathogen because the size is really big. Otherwise, you will learn capillary is also not easy. That something going big things can't go in like bacteria like things. Okay, if bacteria enter, uh, this is like an entry point for bacteria. It's a weak point they can enter to protect. There is a special cells of the immune system allocated. So only option is position these soldiers, the cells of the immune system, to protect this area. And these cells are called macrophages. This macrophages has special name, a Kupffer cell. So they are called Kupffer cells, but they are actually macrophage, right? The macrophage. So they are guard this place. They are guarding this place. So they're continuously looking any threat of pathogen. If the pathogens come, they can destroy. Okay, so that's again very important. The function of the Cooper cell in liver macrophage. They prevent entry of the pathogen as this structure has some kind of a weakness. When it dividing, it make the big holes to make the sinusoid, and the blood is mixed up from portal vein and hepatic vein, and finally all the blood now moving down to the central canal. Then that goes to the vein, the hepatic vein, and then moving to the heart, right? Now, these, in between these cells, this area, the blue color one, this area is the passage for bile transport. This is called passage for bile transport, and this is named as bile canaliculi, like a bile canal. So bile is moving this direction because cells are make the bile, and bile is moving this direction. Okay, I think uh, that's it. We will. I will share this one. Some other pictures that you can understand well. Yeah. So here it made out of the lobe, and lobe made out of this kind of lobule, hexagonal shaped lobule. This is a microscopic picture. So as I told you with the drawings, the central canal and these places like here, they have this all these hepatic artery, portal vein, and the bile duct. And in between the sinusoid and bile canaliculi, we can find in this lobule. So this is a real microscopic appearance. If we convert this to a drawings, This is a kind of a drawing, so you can see a nice a 3D model constructed. So here you can notice hepatic artery, portal vein, and the bile duct. The three structures. You can find it here, right? And that's a central canal, and in between sinusoid and the bile canaliculi. I think the green one try to represent the bile canaliculi, and the blue one try to represent the sinusoid. And this is again another appearance, how it looks like. So these columns nicely make the structure about the hepatic lobule, and here another way 3D picture. So you can also see again these breaks of this the sinusoid, and then macrophage line. So look at this bile canalicule, the green one, bile duct, and the portal venule, portal arteriole, bile duct, the three structures. Sometimes it's called portal triad. Triad is for three: bile duct, hepatic artery, and portal vein. And the central vein, and then the hepatic vein. So all drain to the middle, and then it's going down. Sinusoid and the macrophage. Okay, I think we came to the end. I will upload these files and everything. If you have any issues, personally, you can WhatsApp me to get the password or anything missing notes. Then I can give the password and everything, right? So we're going to meet again next week, and see you next Wednesday again at the same time, right? Okay, take care.
Thank you.
Okay, bye.